All right, I have exams. Okay. Had to cheer you up. All balances out. Good news today. No, it's good news actually. The average was 90. Good. Lots of people got 100 plus. They got extra credit. Very good. I'm really proud of how everyone came through. Of course, you know, there's always a few stragglers. Don't worry about it. But certainly, if you are in the lower category, and nobody failed, but you know, people lower average. Try hard. Your endeavors that people do well. So buckle down and make sure. I will give you the key to the exam later today, so be looking for that. I marked a lot of it, but it's not fully marked with all the answers. Sometimes it was just too much to write. So you'll get the answer, okay? So that's a good study guide for you for the final. The final is comprehensive. This will all come back. Should say, come back to haunt you. All right. Jordan Gilder. Okay.
He said the average was a 90.
Sydney Rudy. Kathy. Okay. All right. Matthew Hart.
Oh yeah, I can see.
Hart.
I think I studied chapters.
Devin Davis.
I'm just gonna go ahead and wing it. Multiple choice. Fill in the blank like word bank. Could be worse. Could be trying.
Were you about to take it?
Caitlin Bam.
I'm gonna say I'll be okay with it.
It wasn't that one.
There's probably some shit there.
Morgan Youngblood.
I did like it, but I wanted to.
Hunter Bright.
You see me left is
Chelsea.
first game.
Zango.
We're asking me questions.
Chelsea. Zango. Chelsea Davis. Matt Smith.
Feels like it's good and warm.
Melissa Hammer. Nicholas Nelson. Someone did well.
Yeah.
All right, Leslie Coates.
What is my love for?
Leslie Harris.
No.
Randy Johnson.
Oh God, I got 20 points on my first one.
Rachel Fletcher. Marla. Is she? Okay. Megan Pitko. Taylor Conway. India Kelly. Chelsea Tiger. Ashton Heterotroph. Richard Moore. Mars. Jenny Soto. Jeremy Francis. Corey Walker. Corey Chapman. Cody Getty. Brianna Milton. Haley Miller. Taylor Chapman. Michelle or Michael, pronounce your last name? Jamaldi. Okay. There you go. Justin Ganowin. Ashley Harris. Greg. All right, so who did not get their exam? You and you are?
Mara Smith.
Mara?
Yeah.
Okay, Mara. I probably pronounced your name wrong, so sorry. Mara Smith, here we go. And everybody else?
I didn't get mine.
So somebody else? You are?
Yes.
Okay, is that everybody? All right. Very good, and I guess Marla's on the way. Somebody has her on their GPS or something. Okay. All right, so, I think just a couple notes on the exam. I think everyone did really well, so that's good. So I think the major things to work on are, when you think about this, is that when I give the keywords, those are really important keywords, right? You have to be able to utilize those in describing everything we do. Second thing is that the processes that we talk about, you have to know them well enough that the sequence of events in which they occur are also important, right? So you can't jump around too much, start off with segregation, only mention part of it, and jump down to septation and back again, because you're just sort of remembering things. In terms of thinking about describing the process, it makes no sense, right? So it gets really confusing. So those are things to work on, the sequence of events. And a couple of you miss the mechanism of penicillin target or mechanism of action. Okay, most of you got it pretty good, but there's a couple of you just make sure that you really go back and think about that. Okay, remember it's just a competition between two structures for the enzyme that we call penicillin binding protein. And then the last thing is, and I can take a lot off of it, but I think the connection is there if you think about it, is that when you're describing peptide or glycan synthesis, right, it goes through lipid 1, lipid 2, gets flipped across, incorporates itself, cross bridges form. Lots of people put cross bridges form based on the enzyme called transpeptidase, but then I didn't really see you call that transpeptidase, a penicillin binding protein, right? That's what it is. And even though we call it a transpeptidase, that is the penicillin binding protein, right? So then when you went to the next question, we've already stated it, right? Because you can say, oh, okay. So that happens at that point in the synthesis process. So therefore, if you add the drug, then there's a competition, penicillin binding proteins. You did a wonderful job there. A lot of people nailed it and said, okay, penicillin binding proteins form cross bridges, and with that connection there, I didn't really see it. A couple of you made that connection. Said, okay, penicillin binding proteins called transpeptidases cause these things, right? And then you went on to the next one. So okay, and this is why they don't work very well. So it was a nice transition. It made sense. Convinced me you knew. So just the little things like that. Make sure you really think about this and put some thought into it. Okay. And the last thing, if I added them wrong, which I can do that sometimes, and you didn't add it correctly, let me know. We'll correct it. Okay. And of course, the very final thing is if you have any concern about how I graded your exam, let me know, and we can go over it again. But I went pretty thorough over it, because I'm pretty convinced that the score you got for each one of those questions is pretty accurate. Okay. Good way to look at it would be based on points. Is that an excellent, above average, average, below average score? And if you look at your overall answer, where do you fall, right? You can sort of see how that's ultimately how the scores occur. Okay, so just think about it. Okay. All right, well, let's move on. We've got a little bit of time today to tackle a new topic. We're going to do some of it today, and we'll continue this on Wednesday. Plan on a quiz on Wednesday. Okay. I know this whole pop quiz thing is kind of out the window, but I just kind of want you guys to prepare for it. And then we have Friday, we have another group presentation, right? So we're moving right along. We're kind of at a very high grade of learning here. So make sure that the two groups, which are going to be groups 5, which word group 5, right here. Okay. So we'll be thinking about the topic, which will start today. And we'll go into Wednesday. But today, I think is a really good opportunity for you to begin to pick a topic. Okay, 5, and then group 6 back there. Okay, so right in here, you guys are the chosen ones. Okay, so be thinking about it, and we'll talk about some things today, and then we'll go on into Wednesday to complete the process. All right. Okay, so what is this term that we use? Called nutritional typing or nutritional types. Well, it's really important for us to understand, just like I did make the correction here. I have chapter 26 on there, delete that. Don't worry about reading chapter 26. Okay, it has to do with fungi and stuff, so we're not going to worry about that. Okay. So really the focus is, if you're looking at these chapters, microbial metabolism, right? Think back to the early days when you learned glycolysis, the TCA, the electron transport chain, aerobic respiration, anaerobic respiration, right? All of those things, we're not going to cover those because I expect you to know the basic fundamental of these. But then we're going to tackle some really interesting questions. In your notes, if you have a chance to look at it already, we'll start off by trying to answer one question. And that's thinking about the types of nutrients that cells use, and thinking about the amount of energy that they can acquire from those. How do you determine that? And there's a lot of ways to do that. You can do that quantitatively. There are some really interesting formulas that you can use to make these calculations. And so that's what we're going to talk a little bit about today. But the main thing is just thinking about an organism, thinking about what it uses as a source of 3 things that really stand out when you think about metabolism. Okay, metabolism is best defined as number 1, the source of carbon, source of energy, and source of electrons. That's really how you should be thinking about it. Okay, so you're making that transition now from glycolysis, TCA, respiration, aerobic, right? So now, a little bit more detailed, so okay, that's microbial metabolism, okay? And so now what we're saying is think about microbial metabolism a little deeper in terms of those three areas: carbon source, energy source, and electron source, okay? So when you begin to do that, then what happens is there's some really interesting terms that one has to know, okay? And there's 5 different types of bacteria that we can begin to put into categories based upon this source of carbon, electron, energy source. Okay, and this table does a really good job defining that. So these terms that we call nutritional types, that's the first order of business that you have to really know. I mean, just sit down and know these. Number 1, know how to spell them. Number 2, know what they are. Okay, because it'll be part of the way in which you describe different types of bacteria. Okay, so you're not just going to be saying, you know, this bacteria likes to grow in this environment, or this bacteria causes this type of disease because it uses breakdown of proteins to fuel itself, right? You're not going to call it this type of bacteria. You're going to call it a particular kind of bacteria. You're going to say this, the chemo organoheterotroph is the organism that's key in terms of defining many types of infectious disease in humans. Okay, and so you'll understand what that means in this section of our class. Okay, so that's the goal that you're shooting for. You have to be able to tell me, using these terms here, what we're describing, because it comes down to the fact that you know what that means, right? So if we go back to that term, chemo organoheterotroph, and that has something to do with infectious disease, it makes sense because number 1, organisms that invade our body thrive upon the different things that our bodies provide. Okay, and in most cases, what's the number 1 source of carbon? If you want to use that as your first example for bacteria that use carbon in our body is, what do you think it is? If you were to take a guess. You got an infection, it's full blown. What's it feeding on?
CO2?
No, not CO2. Good guess. Okay, give me a specific part of the body. Make up that. Organic carbon. Yep. Well, what are they thriving on? You've got an active infection going on, right? You're sick. It's growing like crazy. You're not feeling very good, or you got a huge, massive sore. It's blown into a huge infection. It's not looking good. What are they thriving? Now, in the lab, if you want to grow bacteria, you make up media, put in lots of sugars and things like that, right? But that's not really what happens in the body, right? That's in the lab. So what goes on in the body? What do they just thrive on? That would be a source of carbon. Blood? Not really, but good choice. Good stab at it. What's your tissues made of? If you had a microscope, you could really look at the surface of your skin. Lipids and proteins, right? I mean, that's what essentially cell and tissues are made up mostly of. Okay. So now, if you remember this, and hopefully you learn this in Intro to Micro. If you look at glycolysis, right, and it starts with the sugar, and it's called glycolysis because it uses glucose, and it goes down this process, right, and it goes to pyruvate. Okay, and so you all learn that the source of carbon, is this 6-carbon sugar. And that's the backbone of what most of the cell is made of in terms of carbon, right? It comes from glycolysis. That's how we learned it. So you sort of center on that and say, okay, that must be the main source of carbon. That's where it gets all its carbon, and that has to be it. And that's true. It's huge, and that's how we learn it. But when we talk about infections, if our cells are made up of protein, they also enter and membranes, such as lipids and fatty acids, they also can enter glycolysis as well, right? So that we know happens. Because that if we define infectious disease, then these two actually become the prime source of carbon, because that's what's available to them. So they have the ability to use these. And of course, how do they do that? Well, they produce enzymes that are specific for proteins called proteases, right? Bacteria secrete them, break down these large, complex proteins into smaller amino acids, and then they're taken in, okay? And then here's the TCA cycle. They can also enter here as well, okay? So that's really what they do. The same thing with lipids. These long chains of fatty acids, they secrete lipases, and they break them into smaller pieces of carbon, and then those carbons then enter at various places, and that's what we know as glycolysis, right? So that's what really allows infectious bacteria to grow really well in the body, become full systemic infections that can then cause some serious disease, right? So that's typically what we know happens. So when we think about that, then as Daniel said, we have to now think about how do you describe those kinds of bacteria? Well, it depends on what we're talking about. So if we want to take the first three terms first, let's take a look at carbon. The best way to begin to understand the source of carbon would be really think about the basic backbone of metabolism. Okay? So when we say what's the source of carbon, then the answer would be at what point does it become part of the glycolytic TCA cycle? Okay? And so when you think about it like that, then you begin to understand where the organic examples can then feed into this glycolytic pathway, right? So let's think about the next part of what we would call organic, and that would be inorganic, right? So if we're talking about an inorganic example, then there are examples of what we call carbon sources that are what we call inorganic. As Craig said over here, he said CO2. So I do want to make a little bit of a distinction between how do you define organic and inorganic? What's the difference? How would you do that?
I would say, so organic defined as having like multiple carbon chains.
You're getting there, not necessarily, but it would be.
Wouldn't it have to have carbon and hydrogen to be organic?
There you go, hydrogen. Hydrogen is the key, right? You got carbon and hydrogen, it's organic. So now if you go back to the CO2 as a carbon source, it makes sense now, right? It has carbon and it has oxygen, but it has no hydrogen, right? So that's what we call carbon dioxide, an inorganic carbon source. Okay? And of course, some are more obvious, like nitrogen, right? Clearly, there's no carbon, no hydrogen, so that's an inorganic source as well, right? So that's a real good thing to remember. The definition of organic, inorganic, and the differences are that organics have a carbon and a hydrogen, and the inorganics do not. So we tend to use glucose as our organic example, and we use CO2 as our inorganic example. Okay? And that's kind of what happens here, right? So you begin to look at sources of the carbon. It's either going to be the inorganic CO2 or it's going to be the organic. Okay, because that has to have carbon, and they both do. But in terms of how we define the differences, that's where it becomes one of them has hydrogen, the other one does not. Okay. So that's the first thing. So now we think, going back to the source of carbon, I'm thinking about how are you going to begin to understand what that means. Like I said, it's connection with the metabolic backbone of metabolism, okay, glycolysis. So where would the CO2 inorganics now, their association with that, where would that happen? What's the connection there? How would you think about how the CO2 would now become incorporated into the basic backbone of metabolism? What should happen would be, you have to now think about it in terms of not necessarily backbone of glycolysis as being the beginning part, but you're going to have to think about another system that's a little bit more complex, and it's associated with photosynthesis. Okay. And so now it begins to make sense because, you know, we're all familiar with the fact that we release CO2, plants take it in, right? And that cycle happens. They take it in, plants produce oxygen, we breathe it in, there's a nice little cycle that goes on, right? So photosynthetic cells, plants, photosynthetic bacteria, they have these photosynthetic systems that have the ability to take carbon in. And so there's a process called carbon fixing, okay? And there's a specific type of system that we call the Calvin cycle. Okay, so the Calvin cycle is a good example of where you can now begin to think about how this source of carbon that's inorganic can eventually make its way into the glycolytic process. Okay? And if you follow the Calvin cycle, that's exactly what happens. Single carbons come in, those single carbons become 2 carbons, 3 carbons, 4 carbons, 5 carbons, and 6 carbons. And eventually you now have a 6 carbon molecule that's no different than the 6 carbon glucose that you would start with. Okay, and within that Calvin cycle, it goes through these phases that ultimately what happens is that 6 carbon molecule now becomes part of the glycolytic pathway. And so there you go. So that's the connection right there. That's how you can now enter the carbon source into the metabolic backbone pathways. Okay, through a photosynthetic carbon fixing system. That's how inorganics are defined, the source of carbon. And then the organics simply direct entry into the system by either starting with the sugar, glucose, or proteins or lipids. Okay, and that's how they enter as well. But ultimately, everything goes to that, and that's why we emphasize this so much, that the EM pathway or glycolysis, the TCA, those are critical backbone metabolic pathways that all cells use. Okay, it'll eventually get there. And so when you think about the source of carbon, begin to think about it in terms of how they enter in that way. So that's at least one of the, I think, the best ways to begin to sort of divide how these are different. Okay? The second one is the energy source. Now, how do you think about the energy source? Well, the thing about the energy source is, I've broken these down a little bit for you, is again, there's three types, okay? And how you define energy will matter, right? It will either come from organic, inorganic, so that term inorganic, organic will come up throughout all of this in terms of defining the differences, okay? But then there's a third one that we haven't talked about, and that's light energy, right? And that's connected with photosynthesis. Okay? And we know that you have to have a conversion there. Light's converted to chemical energy. And so that's ultimately the goal of light energy, is that it has to be converted to chemical energy. ATP being the best example, right? It also has the ability to produce what we call gradient energy as well. Okay, and that's used to make ATP. So that's the definition of the source of energy. The electron source. Think about it, there's a pattern going on here. Same thing. It's you're saying, okay, if he's asking me to think about carbon source, energy source, electron source, I need to ask myself, is it organic or inorganic? Because that's the types that are going to be what defines the source, right? Same thing, it's going to be one or the other. Okay? And from that, it then defines exactly what those are. So let's drill a little bit deeper. Okay, so you sort of understand that when you begin to look at the sources, it's either organic, inorganic, or light, right? And then from that, we want to get a little bit deeper and think about exactly how does this happen. So a good question to ask yourself is, you know, where do the different sources enter metabolism? I use carbon as our first example, right? It enters the backbone. Inorganics enter at that photosynthetic system process called the Calvin cycle. Okay. How about the last two? Energy source. How do you think about the source of energy? Well, the source of energy was would be, if you're talking about energy, you're talking about what drives a reaction. Okay? Metabolism is full of reactions, right? So then you need to be thinking about what drives the reaction. You know, in this case, it's either going to be organic chemical energy, like ATP, right? Best example would be the glycolytic pathway. I'm defining it as BEM pathway. Okay. There are a lot of places in there where ATP, for example, is required to drive those reactions forward, right? So that would be a good place to think about it. Because everything goes back to just a few things that we're using as examples that I want you to learn this from. And from that, you can expand it later. For now, this is fundamentally how you really need to begin to look at this. Okay, so you look at the fundamental backbone glycolysis. So, okay, what's the source of energy? Chemical energy, ATP, right? Are there other ways in which this can happen? The answer is yes. Okay, what you can now have is a source of energy that drives another part of the backbone of glycolysis, and that's the electron transport chain. Okay, and so exactly where does that happen? Well, if you remember glycolysis, TCA, those two produce the what we call reducing factors that ultimately drive the electron transport chain that make ionic gradient energy. Okay, and so that's another way to think about the source of energy. What drives the electron transport chain? Okay, and then the third one would be source of energy if it's light, and that's what's going to drive photosynthesis. Okay, so when you talk about energy, just to recap, again, the backbone of how you're going to begin to define this would be the glycolysis backbone, the electron transport chain, or photosynthetic systems. Okay, that's going to help you define what's a chemoorganotroph, what's a chemolithotroph, and what we would call a phototroph. Okay. And then the last one is how you think about sources of electrons. This one has to do with the term reducing power. Okay. So if we say, well, let's ask some questions and think about the source, the electron source. What are we talking about? Well, we're talking about organic or inorganic molecules again. They have electrons, and its ability for them to provide the electrons for metabolism. Okay, that's what we mean by reducing power. Simply means a molecule has the ability to donate an electron. So that's the defining difference between carbon source, energy source, electron source. Okay, so if the question is asked, how would you describe the differences? And if you get to the electron source point, you would say, well, the electron source has to do with a molecule providing the electrons, what we call a reducing power. It donates an electron to metabolism. So that's how you begin to define that. Okay. Because it gets confusing because people begin to think about energy source, electron source, and there are overlapping. It's the same molecule. But in terms of how you define them, it's different. That's what we're trying to learn here. Okay. So electron source, we're defining what donates the electron, called a reducing power. The energy source is what drives the reaction. And it can be the same thing. That's where it gets confusing. Okay? For example, back to energy source, the electron transport chain. It's NADH. It's providing an electron, right? But we're talking about its ability to provide the energy. So if it donates, NADH donates an electron to the electron transport chain, makes a gradient, makes ATP, that's the energy connection. We talk about it in terms of the electron source. NADH is the reducing power. Okay? What happens is it then, through oxidation, delivers an electron. It's a donor. So that's the difference, right? So I need you to go back to your textbook, think about the lecture, think about how we define those three sources. Okay? Carbon source, real easy to remember. Just has to do with the metabolic backbone and what provides the carbon. Okay. It's organic, it's glycolysis. Inorganic is probably a photosynthetic system. Energy source, what drives the reaction? Okay, it can be chemical or non. Okay, and then the third one is what's the reducing power? That's an electron source. Okay, so that's the key piece there. Okay. So let's end. Well, we got some time. Okay, so I thought we were moving on pretty quick. So now, the question that I would like you guys to think about is the amount of energy that cells can acquire from a nutrient. And that matters a lot because that can define whether a bacteria is successful in growing. And if it's successful in growing and it happens to be an infectious pathogen and it's causing disease, then ultimately it's based upon, in many ways, its ability to have enough energy to do this, right? So now, if we think about it, and think about all the inorganics and organic molecules that are out there that are used as nutrients, as food, there's different levels of energy they possess, right? And so the question is, how do you begin to explain that to somebody? Somebody said, well, you've got sugar here, and you've got some sort of inorganic like iron over here. They're both food, but you're telling me that glucose and iron have different amounts of energy. So how are you going to explain that to somebody? And that's what we're trying to learn here, right? So how do you do that? Well, what you do is every molecule has electrons. Okay, it's really interesting about nature. They all possess electrons, but they're in different amounts. So you can take, I'm using ammonia as an inorganic example, and glucose. So they both have different amounts of electrons. Okay, glucose has 24, ammonia has 8. And so that right there, you can begin to think about it and say, well, if they have different amounts of electrons, then I guess that would be one way to explain how they would have different amounts of energy, right? And you're absolutely right. But then you need to tell me exactly how would one come to that conclusion, right? How would you do that? Well, you have to rely on a term called redox potential. So what in the world is redox potential? Redox potential simply means that it's a value based upon energy that's given to a molecule that defines its ability to either donate an electron or accept an electron. And you can either have positive energy or negative energy. This is what we call the redox potential. If that doesn't really make sense, then this hopefully will help you understand that. Okay? These are examples of all kinds of different molecules that are in the cell that we can assign it as a redox potential value, and it's either going to be very negative or very positive. That value in nature defines whether that molecule is more likely to give up an electron or accept an electron. So when you put them together, an event will happen, right? A more likely donor and a more likely acceptor, you put them together, are likely going to allow that transfer to happen. Okay? And then in nature, it just so happens that in glycolysis, aerobic respiration, you think about it, where glucose is your starting molecule, lots of electrons, and your final accepting molecule is oxygen, right? We know that happens in aerobic respiration, right? Goes through the whole process, TCA, NADH, delivers electron, the electron transport chain, it goes down, and the very last molecule that accepts that electron is oxygen, right? Comes to water. Okay? That's what that is. So you can see that the values of glucose and oxygen are given here, negative and positive. And what allows us to make this conclusion that glucose has more energy is that when you take the difference between the energy of those two redox potential values, it's quite large. So number one, it has more electrons, and number two, redox potential value difference between the beginning and the end stage is quite large as well. And compared to what ammonia, less electrons, right? But then when you think about systems that we know exist, ammonia is the starting source of energy, and its final electron accepting molecule can also be oxygen. Okay, lots of soil bacteria have this association. But then that redox potential value here difference is quite small, right? So now you're beginning to see, okay, ammonia, he's telling me, has less energy. These cells that use ammonia produce less energy, okay? And so visually, you can begin to understand this. So, okay, the difference is quite small. So how would you then eventually calculate that final product? Well, we happen to have some really interesting formulas that we can then use to make these calculations. Okay, so this is just a follow through of the numbers that I've given you. Okay, so you calculated the delta G, or you calculated the redox potential value, which is the delta E. Okay, that's the big difference there. You then insert this into this formula. And this is the free energy difference formula. This is key here because this tells you this is your electron number. Okay, so glucose, that's 24, and ammonia, that's 8. You just calculated this by looking at the differences on that table. So now you have two values. You just do the math, and ultimately you get these values that are going to provide you that key, what we call free energy value. And that's basically defines the amount of energy that's available or the amount of energy that's not available. Okay? And so what happens is, when we think about cells successfully surviving, dividing, and growing, there is a threshold. Every cell, on average, has a threshold in terms of having to have a certain amount of energy to support all the systems that allow it to survive. Okay? So what's that energy threshold? It's around 600 kilocalories. Okay? So now it begins to make sense, right? Nature, take glucose, that now it produces 686 kilocalories. It's a little bit above the threshold, so it's plenty of energy for that one molecule to supply the energy it needs to support the systems of a cell, right? It's all good. But if you take ammonia, you take a single molecule, and you look at how much energy you produce, that's 65. That's way below 6. Okay? So that's not enough. It's pretty poor. So then the question is, how do cells survive? Because this is a food, right? How does this happen? Well, think about it. Well, if only one molecule of iron or of ammonia can do this, you're only part of the way. Then how would you make up the difference? You consume more, right? And that's exactly what they do. Okay, those organisms consume a lot more ammonia to maintain that high level of 686, for example. Okay, whereas a respirator that's an organic takes in very few glucose molecules because when they break that down, there's plenty of energy, right? They're good. The other guys are just bringing that stuff in, so the transport systems have to be working at high rate. You got to bring it in as quickly as you can, and then you got to produce the energy so you can get up to around 686. Okay. So whatever ammonia happens to be in the environment will get consumed really quick. And that's the last concept I want you to think about, is that these organisms that we call inorganic utilizing organisms, autotrophs, that use things like ammonia, which is in the environment, that it participates in a cycle called the geochemical cycle. So inorganic utilizing microbes play a huge role in biogeochemical cycle. So make that connection before we leave here today. So the connection is this: the environment, what's out there in nature, full of inorganics like ammonia, iron, sulfur. There's a lot of it out there, luckily, so that organisms that want to use this now have access to quite a bit of this. So they're bringing a bunch of this in, converting it, using it for itself to then grow and be successful. Okay? And the products of those are then recycled back. So now we think about, okay, you know, when we talk about the carbon cycle, we talk about the nitrogen cycle. In this case, that's ammonia, the nitrogen cycle. That's a key part of the ecosystem. And if you didn't have a cell that could bring in a lot, convert all of that in large amounts to something else that can be utilized and cycle back, then the whole geochemical cycling wouldn't be as powerful. Another way to think about it is, what if ammonia, one molecule of ammonia was enough to produce 686? Then it'd be really hard to think about, well, it's only going to use one molecule of ammonia, 1 mole of ammonia. The other ammonia is just going to be out there. Nothing's going to happen to it. The cycle would be pretty inefficient, right? But nature's done it the other way around. They said, okay, well, we're not going to give you a lot of energy from that, so you're going to have to use a lot. But as you do that, you're really cycling this needed ammonia, which carries a critical nitrogen element that every cell needs. And we're going to do it in bulk. So that's another way to look at it. So it's really a powerful link. You're saying, well, why do organisms even use molecules that don't have a lot of energy? What's the point of that, right? I'd rather use something that's high energy. Well, this is what it does. It supports the biogeochemical cycling process. Okay, that's why. So there is a benefit, not so much for itself, but as long as they have access to a lot of these, they're good. It just so happens everything out there in nature's, it's full of inorganics. There's just tons of it. It's a good thing. Okay, so that's one way. If you never thought about it like that, that's why we have them. Okay, and then of course you got the other guys that use glucose. Don't need a lot of it, but when they have access, they grow like crazy. Okay, and we call that infectious disease and other kinds of organisms. And of course they all work together, right? Because think about it, those glucose utilizing guys, they depend on these inorganic guys to do the cycling, because they have to get some of these elements from that process as well. So they're interrelated, right? You got it. They depend on each other. Sort of like an efficient cell, inefficient cell. They need each other to survive. Pretty cool, huh? So that's how you think about it. So anyway, I'll stop here. We'll continue this on Wednesday, and we'll be done. Yes.
On glucose, why is it 24? Why wouldn't it be higher?
Electrons?
Yeah, because you've got the 24 from the carbons, yeah, but what about the hydrogens and oxygens?
Right. Well, it comes down to, when you think about overall molecules, that you can't count the whole thing before it even begins to go through the process of where the electrons go, right? So what they're thinking about is the electrons are actually part of this glycolytic pathway. If you follow those, what's given to that is supposed to be electrons. Right? Does that make sense?
Yeah, so after it goes through the glycolysis, that's...
First breakdown, like glucose and fructose 6, you've already delivered and exchanged some electrons.
Right.
So you just follow the step all the way down to pyruvate, and pyruvate into the TCA. Some of those electrons are going to be given to NADH, so it kind of is in that direction, and some of it's given in the intermediate precursors that ultimately now are going to be used as building blocks to make a larger molecule. So they kind of branch off in all directions. The question is, which electrons are going to be participating in defining the energy source?
Right.
The building blocks, but the ones that make energy. So when you think about it, the 24 electrons when you attach to glucose are the ones that are defining energy.
Okay. So it's not...
energy, but defining energy.
So it's just the electrons that are being put into the energy, not total... Okay. That makes sense.
Okay.
Well, my organic stuff is I think you just go over my